Get started. Yep, we can go ahead and get started. So we're going over the different signaling cascades, which are also called signal transducers, these different pathways, and they fall into one of 6 types typically. Okay, and so we're just we have, yes, it's going to work. The GPCRs, like I said, many of them will have abbreviations, and so I don't want to have to say G Protein-Coupled Receptors every single time. So we have GPCRs, which is the first class that we're going to cover in great detail. And the textbook example is epinephrine, which is adrenaline. Okay? Then we have the RTKs, which are the receptor tyrosine kinases. And the example that we usually used in textbooks that we'll also use is insulin, okay? Which insulin is actually called the bifurcated. What does bifurcated mean, since it's a technical term? Something bifurcated. It's split into 2, like the snake has a bifurcated tongue. Yes, and so it's split into 2. And so there are 2 insulin pathways, but the one that we go into the greatest detail with is the one that does an RTK. Then we have the RGCs, with the receptor guanylyl cyclases. We have the adhesion receptors, which are also called integrins. The gated ion channels, which actually I don't spend a lot of time on the ion channels because we covered that also last semester. You've also covered it in dynamic physiology. It's like the sodium potassium pumps and all that kind of stuff that you've seen, acetylcholine, some of those that are related. And then nuclear receptors. Remember, you're supposed to have a general idea for all of these, give their pluses, I hate saying pluses or minuses, but compare and contrast them. Also, if there's any special exceptions and things like that. All right, so this figure is from your book, and I like to use it as an outline as we go along, and so you'll see it over and over and over again. We're going to right now just focus right here on the GPCRs and go into greater detail. All right, and once again, I apologize if I say secondary messengers instead of second messengers. They mean the same thing. Whenever I went to school many eons ago, we always refer to them as secondary messengers, but secondary or second messengers. Every single GPCR has, at minimum, 3 essential components. Okay? Every single one. First off, they all have to have what's called an effector enzyme. Okay? They are all, and this is really important, they are all membrane bound. They are part of the membrane. So you can imagine something's coming down and it's going to set extracellularly on the outside of the receptor, and it elicits an internal change because of the fact that these proteins cross the membrane. They have 7 transmembrane alpha helices. They're almost all alpha helical. They cross like 7 times. Maybe we went over the different types of proteins and stuff last semester, like types 1 through 7, all that kind of stuff, and if they were polypeptides and everything like that. And so you have to have that receptor protein. It's called an effector. It has to generate some type of intracellular response through a second messenger. We'll talk about some of the examples of those in a moment. Okay. And this is true for many of the different types of signal transducers. But what makes a GPCR, specifically a GPCR, is that it also has to have what's called the G protein. And G protein is short for guanosine nucleotide binding protein. So you can imagine using what it's called G protein. And so, all right, so they all have to have a G protein. Otherwise, it wouldn't be GPCR. Once again, the G stands for guanosine nucleotide binding protein. So what is a guanosine nucleotide? Where have we seen that? Where is that? What else is that used? This is actually where some of these metabolic pathways and metabolic pathways start to intersect and interact. Where have we seen guanosine nucleotides? G. It's not glycine, but G. DNA, right? And so this is related to that. Technically, you're going to find out it's actually RNA, but it's because this is not deoxyguanosine. But yes, this is a nucleotide. And so you're going to find out GTP and ATP. For all of the NTPs are important, but GTP and ATP are used over and over and over again for many processes. One piece of advice, you may want to start doing this, and actually, because normally this chapter is done at the very end of biochem 1, and so we start off with biochem 2. You want to keep like a good half of the battle when we do metabolic pathways, or close to half the battle, is knowing your enzyme classes and families. Okay, and there's several, and so you may want to start to keep like a tally, like a page or something for that. So we'll see some examples of that hopefully later today in the handout that I gave you, and we'll try to keep an ongoing task. And we actually review it over and over and over and over and over again. We actually already know some of these. Like what's the proteases are enzymes that do what? What do proteases do? They break down proteins. So what class of enzymes break down DNA or RNA? Nucleases. Specifically, if it's only for DNA and DNA only, it's called a DNase versus an RNA. But yeah, no, those are nucleases. Okay, so that's what I mean by that, and we'll see these over and over again. Okay, but remember, they all have to have at least 3 things. They have to have this effector protein that has both an external and an internal side that crosses a membrane. They have to generate a second messenger on the inside, because otherwise that external event would never happen, cause an effect on the inside. And then finally they have to have the G protein. Okay, so for second messengers, these are some examples. The one that we're going to talk about the most is called CAMP, or cyclic AMP, or CAMP. I like to say CAMP because it's catchy. All right. But remember, some of the GPCRs don't use CAMP. Some of them use other ones, like calcium. The calcium ion can be a second messenger. Diacylglycerol, some people call DAGs or DAGs, can be. Inositol triphosphates with the inositols can be. I mean, there's a whole bunch of different possibilities. But the one that we're going to go in greatest detail about is CAMP and CAMP. Okay, because that's the one that's used by epinephrine. And I think your book, I'm sure, gives a little table with a whole bunch of secondary messengers that are possible. And we actually did do, let me see. Remember, we had what we call the secondary messenger. Essentially, you have that's crappy. Like, how do I change? It's been a long time. Let's make it where you can see it. Yeah. So we have the enzyme, or the first event. So in this instance here, what we're talking about is we have this substrate coming to bind to the receptor. So here's the membrane. All of mine kind of look like gametes. But you get the idea. That's supposed to be a membrane, okay? And it causes some effect down here, which I'm just going to show as X. And that X would be the second messenger. It goes on and it causes an effect on another enzyme, or more than one enzyme, or things like that. So that's why these would be the second messenger. So in this instance, we're not going to be talking about cyclic AMP. Okay, this is what cyclic AMP looks like. Let me switch back. This is what it actually looks like. It's more realistic. Okay, it is adenosine. Okay, so it is derived from ATP, which is the same one that's used to make RNA. Remember, if it's DNA, this would be hydrogen here, not an alcohol. Okay, however, this is the way many times it's drawn. The oxygen got cut off, but this is the way that many times it's drawn. Just show that it's 5' 3' cyclic group, but this is what it looks like in, like I said, more or less in 3 dimensions. And so that's what it looks like. That's cyclic AMP. It does come from ATP. Cyclic AMP, and I just blatantly plagiarized this from the web. I forgot to put down where I got it from. But this is just to show you from some paper, like some of the effects that cyclic AMP can have. You can do bronchodilation. We talk about, prevention of aggregation. It's used for, I don't remember, obviously this was talking about some drug specifically against glaucoma and things like that, reduces blood pressure, anti-inflammatories, so on and so forth. There's lots of different physiological responses that ultimately came from cyclic AMP, okay, which should make sense since, CAMP is used for epinephrine. It's the second messenger for epinephrine. And what is epinephrine used for? It's a hormone, but what is it? What is it? Like you learn about in AMP or in biology, you hear about it. What is it? They like to give a little fight or flight, you're running from the bear, which I always say, and then I give you a little thing that, you should run downhill from bear, not uphill, but this little factoid for you for Jeopardy. But yes, and so it should make sense, bronchodilation, anti-inflammatory. You don't want to also be getting inflamed as you're running from the bear and that kind of stuff like that. Okay. All right, so now we're going to talk about, it's called the beta-adrenergic receptor system, but it's the one for adrenaline. Okay? And this is just the textbook, quote-unquote textbook example. There are other GPCRs. This is just to show you a specific example. All right, so the receptor it has is the adrenaline or the epinephrine receptor. Then there are terms that get used a lot of times here called agonist and antagonist. If you took medicinal chemistry with me, we use that term a lot because agonists means it can bind the receptor and it has the same effect as a natural ligand. Antagonist means that it binds, but it doesn't. Essentially, it blocks whatever normally binds there, and it doesn't elicit the second messenger effect. So you can think of histamine receptor. You take an antihistamine like Benadryl, it can bind to that receptor, but it doesn't cause that second messenger to go off. Okay, and so therefore, your allergies are better, which many of us can relate to. So, but yes, whereas an agonist would be a histamine derivative, which I don't know, like I had to go in for allergy testing actually multiple times recently, and I don't know if has anyone in here had gone in for the skin pricks and stuff like that for allergy testing? First of all, very uncomfortable, but secondly, they have to give you a positive response when they give you a histamine, and they deliberately do it, and so, you swell up really big, really fast, okay, in that part of your back or your arm that give you the histamine. And they give you a negative one, too, with saline solution as well. So histamine would be an agonist. The antihistamine would be the antagonist. That's just an example here. But not in this that's for the histamine receptor. Obviously, here we talk about adrenaline agonist or adrenaline antagonist in this instance. All right, so this just shows you examples. This is what, and hopefully by the end of the semester, God willing, we have enough time, we actually cover epinephrine or adrenaline in greater detail when we do hormone regulation and the hormone metabolism. But that's epinephrine. We can say this is isoproterenol, which is an agonist. When we have propranolol, which is an antagonist, this just gives the relative KDs. So we can see that this antagonist binds much, much, much better. Like what is that? From 3, about 1,000-fold tighter than epinephrine. Whereas this one only binds about 10-fold tighter. Okay, so let's just give some examples. Or if you're reading anything towards your papers or just in general whenever they talk about agonists or antagonists. All right, so this is the handout that I gave you. And we're going to many times, sometimes I ask people to essentially draw this and to take it step by step, or at least be able to explain it. And hopefully by the end of today, it'll start to make sense. Like what happens at each step. You can take notes. I used to have I didn't give a name called VARSCAT. I can't remember what it stood for, beta-adrenergic. I can't remember now, but it was like back when the class was bigger and we invited into two and they had multiple groups and they would try to compete for some extra credit points, which was the extra credit you want. All right, so in the beginning, for fake integration. No, what must have to happen before anything can happen and you see that bear coming towards you, what's the very first thing that must occur? Very first thing, right here where the arrow's at. And so, and this is true for every single GPCR. The very first step, you have to have that signal that binds to the specific receptor. If it doesn't ever bind there, it's never going to happen. That's why sometimes this is really, really simple things. And so you have a binding event on that outside of the GPCR. Here we have epinephrine or adrenaline coming in and binding. Okay, now if it's bound, that's one thing, but what must happen next? In order, the moment that it binds, there must be something. Even before it can elicit the second messenger. You have a it's a conformational change. I like to say that, that tells you but something that binding of it here causes some type of conformational change that's going to have an effect on this other side. Okay, and here what it is this is where the G protein comes into play. That's what this is. The G protein here, which to me kind of looks like a liver, like an actual liver. There are 3 parts to it, alpha, beta, and gamma. And G proteins always will have, I mean, assuming you're healthy, always have some form of guanosine, at least one guanosine attached. It could be GTP or it could be GDP. Okay, so that's what G proteins do. They have a binding site, they have at least one binding site. It has to have either GDP or GTP there. What happens here, and they give them, they give them special parts. There's the big GS, which has the alpha part, and then there's I can't remember what the G beta and G gamma called there. But so what happens there is the moment that conformational change occurs, it allows and it kicks off the GDP, and it allows GTP to come and bind to the G protein. When the GTP binds to it, now that GS is active, that alpha subunit there, that's all active. Okay.
Is this next step worked out once for the extra?
It was at that time. So what do you suppose happens when it becomes active? And it's kind of, they're trying to show that in the picture here.
The secondary message now, or is it still?
No, we're not there yet.
Oh, wow.
I know. It's going to eventually be a second message.
Is it like a migration?
Yes. So what happens is you can see it's like the alpha and beta gamma subunits separate from each other. And so that GS alpha, that part, actually translocates and moves over to AC. Okay? And I don't want to tell you what. Okay, why? But this isn't quite. Well, actually, I can. Go ahead and show. Oh, whoops. It helps you if I show it on this one because they're not connected to each other. Okay. So it translocates. It moves over. Because it has GTP. That's what this picture is showing. It's got GTP on it because that initial binding event by the epinephrine. Remember, epinephrine binds, causes a conformational change, kicks off GDP from the G protein, the alpha subunit of the G protein, and it allows GTP. When GTP is bound to it, now it's activated the G protein. G protein moves over to adenylyl cyclase, which if you have a sheet of paper or something where you're doing, we've already talked about proteases and nucleases. We have another class of enzymes called cyclases. So that's important, somewhere to write that down. We have cyclases. Okay. What do you suppose cyclases do? They could break down cyclic things, or what else could they do? They can make cyclic things, but they either make or break something that's cyclic, okay? All right, so what happens here, and this little arrow that's down, it's right here. It's adenylyl cyclase. What does it zoom in from organic chemistry? What does that YL typically mean? Like hydroxyl and methyl and all that. It means it's attached to something anyway. No, that's part of something. All right, so this is it's always hard for me. Wait till we talk about uridylylation. That one, adenylation I can say, but the one with U is like called uridylylation. And so it's difficult to say. But with A's, it's called adenylation. So this is adenylyl cyclase. So what that little box with the exclamation with the question marks on it, that's showing you a reaction that's occurring. Okay. So in order for this to all come across, it utilizes another GTP. Okay, this is not the same GTP. It's called it's a hydrolysis reaction in order to get it to move.
Because the tri part becoming di.
No, it's still tri. So that's why I always told you it will always bind at least one, if not two. So this one here, it's got two different sites for G's. we have the site that's right here that says whether or not it's active or inactive. Whenever it's GDP before the epinephrine binds, it's inactive. When epinephrine binds, it causes some type of conformational change. It allows a GTP to be there, and therefore it's active. However, in order for this to move, it requires an energy source, and so then we do it. There's a hydrolysis there that goes on. Okay. So this is just I didn't give you this handout because I didn't have it already blocked off. But this explains the entire process to blow up the bed. And so what we can do is now we can just follow along what happens whenever the GDP is initially bound. It's inactive. GTP comes in, activates it, and kicks off the beta gamma subunit, which can go back to wait till the thing's everything's finished. Okay? But then what happens is you have to have some type of hydrolysis that is occurring in order to ultimately activate the adenylyl cyclase, which is the next one in the process. Okay? Remember from last semester, the PI is inorganic phosphate. And so then once it's been hydrolyzed, it's GDP. GDP then goes back, and now you're ready for the next epinephrine to come along. Okay?
So that's step, the GDP, and then the hydrolysis is step 2, where that GTP becomes GDP. The phosphate that's being kicked off in that process, is that what's attaching to the GDP to make it GTP? Is there like...
No, I think I understand what you're saying. So no, this is this was the original GDP. Maybe you can think of like the time before. And this is the new one that comes in and kicks off the old GDP. And the old GDP goes up. It can actually be a second messenger for an indicator for other things.
So is it like replacing it?
So it's replacing it. And then when it gets to adenylyl cyclase, it has to activate the adenylyl cyclase. Mm-hmm. And the way that it activates the next, that enzyme, the adenylyl cyclase, is another hydrolysis reaction. So it kicks off that phosphate, so we have a new GDP, which goes back, since it's no longer active, it travels back to the receptor, connects with the beta and gamma.
This GDP was made from that GDP that came in, basically.
This GTP, I think.
Right,
I think
so
you said GDP twice.
So this GDP is essentially the GTP that came in instead of...
Right, ultimately. Mm-hmm. And so as far as constantly kicking, it's not a futile cycle in that sense. And this is one of the reasons why I also want to put this. Oh, and this whole enzymatic activity that's occurring with G proteins is called GTPase. Because they put the ACE ending on it. All GTPase activity, and that's what this means right here, see GTPase, that means it can hydrolyze GTP in specifically. Okay? And so that's why they'll talk about papers when they talk about GTPases or something like that, but that's what that means. And I just want to point this out that cholera, you hear about, what happens with cholera? What's bad with cholera? Cholera, there's a cholera toxin that comes in. And what cholera toxin does is it inactivates the GTPase activity completely because it literally forms a new bond to an arginine that's important. And so therefore, since it can no longer inactivate the GTPase activity, when it's bound to the adenylyl cyclase is always turned on. It's always turned on, and you're going to find out what adenylyl cyclase does in a moment. Then that means that the signal keeps going on, and it doesn't get shut off. Okay, and so that's why ultimately you can die. Right? You can get diarrhea really bad with cholera, and there are other bad things that can happen with cholera. And so it's almost essentially that your body is running on adrenaline all the time. You can think of it in the simplest terms like that. Okay. So, all right, so now we're going to be talking about this next, what's occurring here, which it's AC, which I already know, I used to put it in as hopefully it's a freebie, but AC stands for adenylyl cyclase. All right, so now we're talking about what happens with adenylyl cyclase. So we have the GTP come in, it's activating the adenylyl cyclase, and it's going to continually do so unless it gets hydrolyzed, it has its own activity and goes back. So that's why cholera blocks the inability for the G to go back. Okay, you can think of it there, you get stuck in this position. Okay, so now, which I did, that's the second one I do believe. There. All right. So now we're at adenylyl cyclase. So what do you suppose that compound is? This is what's happening in adenylyl cyclase. What is that compound?
Same triphosphate. ATP.
It's ATP, right? So it makes sense. Adenylyl cyclase has to do with ATP, and so that's ATP. All right. So now, pardon?
Oh, I was just guessing the enzyme,
And so, does anyone remember what does PPI stand for? It's inorganic. Well, they give it a special name for the P. It is a diphosphate. But what's the name that they give it? It says it's called pyrophosphate, but it is a diphosphate. It's inorganic. Okay, yes. So it's inorganic pyrophosphate to make this compound right here. Okay, and so this enzyme that goes and takes ATP is none other than adenylyl cyclase. Don't overthink it. Because this should make sense now. It takes ATP, which is an adenylyl group, and it cyclases it. It makes it cyclical. Okay, and so that's why it's called adenylyl cyclase. And this compound, once again to review, what's the compound called? It's cyclic AMP. It's CAMP. Cyclic AMP. Monophosphate. So it's CAMP. So this is where, this would be the end for adenylyl cyclase. Okay? Because this, once again, this would just, if this kept on, that's really good. It just kept on happening, you'd have color essentially, you'd have the bad side of this. So you have to have some way to break it down. So this is some way, the way to turn it off eventually. Now, CAMP itself goes on and does a whole bunch of other things. And then we have to talk about how to go ahead and break it down. Okay. So we're going to actually skip the enzyme here to say what's the name of this. It goes from cyclic AMP to, yes, which is called AM. Right, it's AMP. Don't overthink it. Okay. All right. Yeah, this is worth the devil. All right. Okay. It is a type of hydrolase, but that would be like, I would give you most if not all the credit for this one. This enzyme is very important. It's got a generic name, and usually that's important in the sense that those with generic names typically are more promiscuous. So you see them multiple times. What kind of compound is this?
No.
Don't overthink it. It's cyclic. Okay, that's one of the names of this. That's the first word. So it's like, sounds like first word. Cyclic. Okay. What else is that compound? Not only is it cyclic, but it is.
Aromatic. No.
What kind of biomolecule is it? What is ATP? What kind of biomolecule is ATP? It's a nucleotide. So this is cyclic nucleotide, right? And then the enzyme itself is because if this was in DNA, just don't ignore this part. And what kind of bond do they say? What kind of backbone is it? The DNA backbone. Phosphodiester. Okay, because and it's called that because this is a phosphate ester. It goes phosphoryl group O. And so this is cyclic nucleotide phosphodiesterase. That name tells you exactly what it does. It also lets you know that it most likely could take like CGMP and do the same thing, or CCMP, or something. You may not have the same affinity, but so cyclic nucleotide phosphodiesterase. That should make sense. That literally means it takes a cyclic nucleotide and it breaks apart that phosphodiester bond. So now you can. I'm going to cover that in more detail later on. And so that's why you don't see the phosphodiesterases as often, but I mean, that technically is, those are types of bonds. Okay. Types of enzymes, not bonds, I'm sorry. All right, so now we had cyclic AMP. This technically is the second messenger, the secondary messenger. We already talked about how you can break it down to keep the signal from going on. But now cyclic AMP, if you want the signal to go on, it goes here, step 5. This is really important. What do you suppose happens when we have our generic, which I don't have enough room to draw there. Remember we had our signal, which I'm just going to call S. S bound to the receptor in the membrane, and it turned on the secondary messenger. What did that secondary messenger then go on to do? Don't overthink it. It amplified it, and it amplified it by doing what? I think that there's that one picture that we had early on. How did it amplify it? Don't overthink it all. This was, like an enzyme, the very first enzyme within the cascade. It goes, we have our second messenger, the second messenger then goes on and does what? How do you amplify something? What happens here? Yes, you have another enzymes, okay? Cyclic AMP is very specific in what enzyme it comes in and activates. So all it does is it's going to activate the next enzyme in the cascade. That next enzyme is called, and we're going to see this enzyme over and over again, it's called pKa. It also has a very, very generic name. I mean, very generic name. Okay? That pKa does stand for something. Does anyone want to guess what P stands for? Phosphate or phosphate, makes sense. It's protein. Okay? It's protein. The K stands for kinase, which is, if you're writing down, having a list of enzymes, we're going to see this class of enzymes over and over and over again. Okay, and then the A stands for A. Okay, because it got turned on by cyclic AMP. So that's why protein kinase A. What protein kinase A does, or kinases in general, I didn't write it out with this one. This has to do with the transfer of phosphates. And I'm going to abbreviate phosphates that way. I can P with the circle in it over and over again. Transfer of phosphates using, and this is the important part, using ATP or GTP. I guess technically it can be any of the NTPs like TTP, UTP, whatever, but these are the two. Vast majority are ATP, but there are some that utilize GTP. This is important, okay? And the reason why is that's not the only way to transfer phosphate. You can transfer inorganic phosphate as well, but the enzymes that do that are called phosphorylases. So these transfer phosphates without using, an NTP of any sort, like ATP or GTP. Usually they're the ones that transfer phosphates, inorganic phosphate. Okay. So that's why, comparing contrasts. So we're going to see this. That's why pKa tells you it's a kinase that got turned on, that got activated by CAM. That's what the A stands for. What is it? My old boss hated when we use like kinase as a verb. What is it kinasing, you suppose? What does the name says that it's kinasing? What does the P stand for? Proteins. It adds phosphate groups to gazillions of proteins. Okay? So that's why it has a very generic name. Many times you're going to find kinases that have very specific names, or more specific names like hexokinase, which is only kinases, sugars that have 6 carbons in it. Okay, so whereas protein kinase A, it phosphorylates using ATP, lots and lots and lots of things. All right, I wouldn't expect you to go over at this point in time. This just shows you that it in and of itself is highly regulated, and how it's activated. It's got another protein called ACAP, which is a kinase protein. But what happens is CAMP comes in and it binds. Whenever CAMP binds, it releases the regulatory subunits. Okay, so now the substrate is all the other proteins can come in and bind it. All right, so finally, what happens here is we've got the phosphorylation of lots and lots and lots of proteins. That's what causes the physiological response, okay, that you see from epinephrine. CAMP didn't do it directly. Even the G protein itself didn't do it directly. G protein ultimately goes and turns on adenylyl cyclase. Whenever adenylyl cyclase is activated, it takes ATP and makes it to CAMP. CAMP then goes and it activates protein kinase A by releasing the catalytic subunit of it. Protein kinase A is the enzyme that goes on and it phosphorylates lots of proteins. It phosphorylates lots of other enzymes, and it's the big powerhouse for the epinephrine pathway. Okay? And once again, this would continually happen unless we broke down the CAMP. If we can't break that down, then we would die because of the fact that you would always have fight or flight going on. And so it was broken down by cyclic nucleotide phosphodiesterase. Okay. So just to show you some of the examples here, and we're going to see this and talk about pKa a lot and all these pathways. And it should make sense what these do. Why think about running from a bear? Whoops. Okay, this is just one example. Glycogen synthase. It's regular, and it shows you, and it should make sense knowing, like a phosphate. Phosphate itself consists of phosphorus, and what other element is in phosphate? Oxygen. And so the things that get phosphorylated typically are going to be serines or threonines because they're the ones that have alcohols on them. So that's why if you notice the S is in a different one or the T is in a different color. And it just shows you the part that gets recognized and gets phosphorylated. Okay, but we have glycogen synthase, which is what makes glycogen. Some things get upregulated and some things get downregulated. So if you're running from a bear, you're not going to want to make more glycogen. You want to break it down, so you're going to actually downregulate that one. We have the phosphorylase kinase, which now knowing what you know now, kinases, what do they do? That class of enzyme, what does a kinase do?
Phosphorylate through the use of ATP.
It phosphorylates using ATP or GTP. And this one, phosphorylase B kinase, what do you suppose it's going to be kinasing? It kinases phosphorylase. Like that's why I tell you enzyme names, many times, not always, but many times, they tell you exactly what they do. Or what, sometimes they have more than one function, but they give you the idea. Literally that means it adds a phosphate group or can remove a phosphate group depending upon, well actually this one adds a phosphate group to phosphorylase, and phosphorylase is involved in the phosphorylation reaction. So that should be, because it's an enzyme in and of itself. All right, we have pyruvate kinase, which is actually part of glycolysis, which gets turned on whenever by pKa. Which makes sense because it's the last step for glycolysis, and so therefore you want to make sure that you break down your sugars even more to get more energy as you can. So what do you suppose, knowing what we know now, a kinase, once again, if you keep saying it over and over again, it's going to stick in your mind. Kinases do what? They phosphorylate things using ATP or GTP, and this one also uses ATP. And what do you suppose that it's going to be? What other compound do you suppose it's actually involved with? That's pyruvate. And we're going to actually find that this is one of the exceptions. It's actually named for its backwards reaction. But yes, we'll talk about like dehydrogenases later on and lipases. But you can see things over and over and over again. For those of you that are in chemistry of cancer, they're the histones that are activated. And so therefore, it allows for more rapid transcription. It gives the polymerases more ability, easier to make it for you to do transcription to make more proteins and stuff like that ultimately. So that's why you can see it over and over and over again. And that's that same figure right there that it's shown before. Okay, so I just wanted to show this because it's amazing. So if we look here, which is kind of hard to say, it just says X molecules, okay? So, you know, for example, if there's 1 or 2 or 5, what have you, but there's X number of molecules. We have a single binding event. And this is hepatocyte, what does that mean? If it's a hepatocyte, it's a cell from the liver, and we're going to find out liver does a lot of things. So this is in the liver. It activates, you know, the GS alpha, which goes on and turns on adenylyl cyclase. So 1 ATP becomes a cyclic AMP. This 1 cyclic AMP can then go on to make 20 molecules. What it does is it activates pKa. Now we have 10. That single binding event now has activated 10 pKa's. pKa then goes on, and this is just one example, to do phosphorylase B kinase, and it activates 100. Then that activates glycogen phosphorylase 1,000-fold, which goes on and it breaks down glycogen to a single glucose 1-phosphate to ultimately make glucose, but it does it 10,000 times. So that means for every single epinephrine bound, 10,000 glucoses get released. That way you can run from the bear. Okay, and this just shows you that epinephrine is not the only one that does it. These are some of the other signaling cascades or signaling transduction systems that utilize CAMP as well, including our arch enemy, histamine. No, histamines do have importance. It's just the fact that they're usually out of whack. And so, okay, are there any questions over this? These are the that's just an example of GPCR. Remember what the 3 main things that they have. You have to have an external signal that binds to a membrane-bound protein. You have to have some type of G protein. You have to have an elicit an internal effect. Okay, do a second messenger, I should say. Okay, then I'm going to go ahead